Okay, we're back again. This time to talk about immunity again, but this time we're going to talk about specific or adaptive immunity. And this is different from innate in that you are not born with this. This is something that you develop in response to seeing an antigen of some sort. So, specific immunity is the body's ability to recognize and defend itself against specific invaders and or their products, such as toxins or different chemicals or, even eggs or something. So we have what's called memory, not this kind of memory, but antibody or immunity memory, which allow it to respond quickly to a second encounter of the same pathogen. The study of all of this, innate and specific immunity, is called immunology. Some of you may be interested in studying about that. It's a pretty broad field, much broader than, back in the 70s, it was kind of new, but now it's huge, and they're even looking at immunity to fight types of cancer and all that kind of stuff. So here's the chart or the org chart, if you will, of the immune system. We talked about the things on the left-hand side, which were the innate immunity, including cellular and humoral components. And this time we're going to talk about the adaptive or specific immunity, which is the second line of defense. And it also has cellular and humoral components. So cell and liquid components. This is it again, adaptive or specific with cellular and humoral components. So, some hallmarks of specific immunity are that it is acquired over time. So, when you're born, you don't have any antibodies, you don't have any specific immunity. You have a little bit that you get from your mom, from the womb and then also from breast milk, but very little. So the majority of it is acquired over time. And it includes various cells, tissues, and organs. They're all part of this specific immune system. We saw in the last one that there was blood cells and different chemicals. And there's even more involved in specific immunity. But the primary thing that happens is that an antigen triggers a very specific immune response. It's not just general like the innate system was, it's very specific to that particular antigen. Okay, so what's an antigen? We talked about this last time. That's any molecule or foreign body that triggers a specific immune response or any kind of immune response. That's an antigen. In specific immunities, it's a specific immune response. I'm sorry I keep, hashing over that or repeating specific, but I want to make sure you understand that there are some differences in talking about specific. So, some things that can trigger a specific immune response are cell walls of bacteria, capsules of bacteria, pili, we talked about those, flagella, all of those things can cause an specific immune response. And then also proteins of fungi, viruses, protozoa, and those kinds of cells as well. Food and dust can also be antigens. Some of you who have seasonal allergies know that for sure. And antigens, just like in innate immunity, are going to enter by various methods. So you'll have a break in the skin. They might get through your mucous membranes when you're breathing or as you swallow them. They can also be by direct injection. So with a bite, like from a dog or a beaver or whatever else you might get bit from, or a needle. In the case of blood-borne infection, you could get it from a needle. And then also organ transplants and skin grafts from different people can have different we have markers on all our cells, and that's how we know we are we, and something else comes in is not us. And when you're getting an organ transplant, the person who is donating that organ has different markers on all of their cells than we do. And so that's why it's difficult to do transplants. You have to take immune suppressing drugs for pretty much the rest of your life because you're going to form a response against, let's say it's a kidney, and you're going to try all your cells are going to try and kill that kidney because it's not you. And so we have to give you immune suppressing drugs so that you won't mount a horrible response against that. When they talk about COVID and immune suppressed individuals, well, that's often why. You could be on chemotherapy from cancer, or you could be on immune suppressive drugs because you have a transplant of some sort. And lots of people have transplants. I mean, they don't have to be, like heart and lung transplants. They can be, cornea transplants, kidney transplants, all kinds of transplants. So a lot of people are on immunosuppressive drugs. And then skin grafts. If you're a burn patient, sometimes you'll get a skin graft from a donor patient or even a cadaver. They'll use a skin graft from another individual. And then also, if you get a blood transfusion, there could be antigens on those blood cells that you're unfamiliar with. When I mentioned cornea. So I'll tell you one other thing about cornea. It's like, you don't think of cornea as being, I mean, it's a lens basically that's on your eye. But there was a case of a person who got a cornea transplant, and the person had died from rabies, and the recipient then got rabies. Bizarre, huh? So that's why they test everything now really carefully. All right. So I mentioned anything that can get into your skin. So, you could get a bite from a dog. You could have surgery and have a wound and bacteria or non-sterile instruments or someone coughing or even yourself. You can rub something in there or from the water, who knows. And then on the right-hand side, we have there some bacteria, different kinds of bacteria, and those can also get in there. Excuse me. So the specific immune system is made up of different components, and one of the primary ones is your lymphatic system. We talked about lymphocytes in the last lecture, so this is the overarching part of the lymph system. So the lymphatic system is an extensive network of vessels, nodes, and ducts through almost all your bodily tissues, very similar to your circulatory system, but it has lymph fluid rather than blood. And that lymph fluid is very important. So your lymphatic system is composed of lymph fluid, lymphatic vessels, and lymph nodes. You've probably all had a little bit of a swollen lymph node here when you've had a cold or the flu, and also lymphatic cells, of which lymphocytes, blood cells, are part of, not the only lymph cell, but one part of this. And your lymphatic fluid and your whole lymphatic system screens your body for foreign antigens constantly. It's a one-way system. That lymph is in your tissues, and it kind of drains into the lymph vessels, and then the vessels carry the lymph fluid past all your lymph nodes. So, if you're draining here, your lymph fluid in your nose and your throat and stuff is going to drain into these lymph nodes, and then they will filter and hang on to any antigens and kill them if they get into your lymph node. So lymph is liquid. It's very similar to blood plasma, and very few cells in this, but there are some cells. And this arises from fluid that's leaked from your blood vessels into surrounding tissues. Okay, so in your circulatory system, you have blood, and some of that fluid goes into your tissues, your cells of all your body, and then that becomes the lymph fluid. It's not blood anymore. It's considered lymph fluid. So if you get a wound, then more lymph is going to leak out of those cells that have been involved in that wound. And this lymph fluid will carry the pathogen to your lymph node to be filtered and or killed. Okay, so you get a cut, and let's say it goes all the way down to the lymph node in your armpit, that the armpit is going to filter the pathogen out. Let's just say it's a staph bug or something, and it kills it, or at least filters it. So this is a diagram, kind of rudimentary, of your lymphatic system. And you can see that there are a number of vessels. And then you also can see the purple kind of roundish structures are lymph nodes. You have a lot more than that, but this is kind of the basic of it. You also have a thymus and lymph nodes, I mentioned. Your bone marrow is also part of your lymph system. You have a spleen, which is a huge part of your lymph system. That's going to filter out like dead blood cells and other things. But all of this lymph fluid that flows through this lymphatic system is goes through a certain lymph node, one or more, and then it flows back towards your heart and eventually is recirculated. All right, you have about 500 to 600 lymph nodes. Now, the ones you probably know about are usually the ones right here because those are the ones that we feel enlarged most commonly because what's the most common disease we get? It's probably the common cold. I mean, I think most people get a cold at least every 2 or 3 years. Some people even more than that if you have young children, especially because they're bringing lots of stuff home. Anyway, 5 to 600 lymph nodes. And these lymph nodes hold lymphocytes, and those lymphocytes recognize and attack any foreign antigens that get into the lymph. And the lymph nodes receive lymph from the lymph vessels. So the lymph fluid is flowing along, and then it gets to the node, and the lymphocytes in there are like, whoa, let's get rid of this junk here. So here's another example of lymph nodes. You have the cervical, which I mentioned are here, but they're also all along here. And then you have groin, you have armpit up here, and you have abdominal regions. So it's a really good system that pretty much covers the entirety of your body. And again, when we have 5 to 600 lymph nodes, they're not all going to be diagrammed in one kind of cartoon image. The tonsils and the mucosa associated lymphoid tissue in your mouth, those are all part of the lymph system. Some of you, they rarely take tonsils out these days, but some of you may have had your tonsil out, and that's because they were filtering and they were filtering really well, but unfortunately they kind of got infected with the organism and then got enlarged and you had trouble breathing or whatever. But now, like I said, they rarely take tonsils out. They just try and treat the infection and allow your tonsils to remain because they are important in screening out for the rest of your life any other foreign antigens that may come in through your mouth or sometimes your nose. So here's a diagram of what a kind of a cartoon diagram of what lymph nodes look like. You can see the armpit lymph nodes there. They're spread out kind of all over, and those in that case are kind of swollen, so you can see them a little bit better. And then over on the right, there's an image of enlarged lymph nodes in the two little dots there. The green is a system of lymph nodes. Then I mentioned we have lymphoid cells. One of those is the lymphocytes, and those are the smallest of leukocytes. So you might think back to when I was showing you the images of different types of cells, and the red cells are pretty small compared to the white blood cells. And here we can see that the lymphocyte is about the same size as a red blood cell, so very small. So there are two main types of lymphocytes. There are T cells and B cells, and those travel through the lymphatic system. Okay. And at the nodes, they come in contact with viruses, bacteria, and foreign particles that have come through in the lymph fluid. So they're packed with lymphocytes. Your nodes are packed with lymphocytes, and they also circulate. Okay, so when a foreign object comes into your lymph nodes, then that's when the lymphocytes kind of take over. Following contact, a lymphocyte is going to form antibodies, and they can produce antibodies from memory if they have previously encountered a pathogen in the past. It is a highly specific and long-lasting response. So when my kids were little, I got a number of infections, right? Number of colds and a couple of flus and whatever, lots of infections. But after they got to a certain age, then I stopped getting infections quite so commonly. And so did they, of course, at a certain point. But, and now with my grandsons, I don't usually get their infections. And the reason for that is because I have seen that particular virus before, and my body has made memory cells or memory antibodies that recognize it the second it comes in, and they go crazy on it and take care of it before I even really feel anything. Okay, so let's talk about what that response is. So we're going to talk about antibodies. That's what keeps us healthy. So antibodies are molecules that are made by our body that bind antigens that get past our immune innate defenses. Okay, so something gets in through your mucous membranes, they get in through your skin, they get in through your eye, who knows? Antibodies are also called immunoglobulins, okay, and it's abbreviated IG. And there's several different types of antibodies that show up at different periods of time, but we're not going to go that specific. We want to get into the plagues. All right, so antibodies have specific binding sites. Now, if you think of anything and everything that can ever get into your body, theoretically, we can build an antibody. We actually have an antibody to that particular thing. Anything. I mean, there must be billions and billions of things that we can make antibodies to. And theoretically, we have a cell that's going to recognize that. But first, let me describe this in a little greater detail. So on the left-hand side are antibodies, and they're got a little bit different shape at their binding site. And the binding site is where the antigen, where the antibody is going to attach to the antigen. Remember, the antigen's coming from outside, and we're making the antibody. So in the first case, you can see that that antibody has bound to kind of an oval-shaped antigen. And in the second case, it's a round binding site and a round antigen. And in the last case, we have I guess it's an octahedron, six-sided antigen, which has attached specifically to a specific antibody. Okay, so binding site is very specific, and theoretically, we have one that will fit anything and everything in the grand design of things. So what is the antibody function? Well, the first function, as we just saw, was to bind to antigens. So when it does that, it, well, first it's going to, it's, the first job is like there's one antibody, and it's going to bind to an antigen. And then when more start showing up, it's going to agglutinate. So all of those different little antibodies are going to bind to the antigen, and then they're all going to form a bunch of cross, like a spider web or something. And so pretty soon it's just a clump, right? Theoretically, and if we don't get sick. And then it's going to neutralize that antigen. So the antigen cannot perform whatever function it is, whether it's sending out chemicals or it's going to invade a cell or whatever. But the antibody should neutralize it. And then the antibody is going to stimulate chemicals, our own chemicals, phagocytosis, and inflammation. Okay, so that's our antibody. Now, where do antibody comes from? Oh, let me go back. This is just a graphic of what I just described. A on the left-hand side is agglutination, and you can see that antibodies have attached to the antigens, and they're creating a little clump of cells. And so those antigens, bacterial cells, aren't able to do anything that they want to do, even if they have flagella. All right, and B, neutralization. So when the antibody attaches, then the bacterium or the virus or whatever can't perform its functions. And then finally, on the bottom, we have what's called opsonization. And basically, that's just when the antibody is attached and the phagocyte recognizes it as something to get rid of. So that you can see the phagocyte ingesting that particular complex, the antibody and the antigen. Opsonization. All right, so here we have an antibody that recognizes an antigen and it attaches, right? Okay, so if we have billions of these and each one is specific to one antigen, how are we going to get along with just one antibody? Because if you get infected with a cold, you have millions of viruses that are coming into your body. Or if you get a bite, there's millions of bacteria. And if we only have one antibody for that particular antigen, it's not going to do as much good because we're going to have like 5 million more antigens coming in. So what happens next? How do we overcome that problem? Well, we have memory cells that remember when they have seen an antigen before. And that's why you are less likely to get something after you have been exposed to it. In fact, there are many things that once, in fact, most things, if you have made an antibody to it in the past, then you're not going to get it again, or you won't get it nearly as severely because these antibodies remember and go on the attack right away. All right, so an antibody triggers an immune cell. Remember, there was T cells and B cells. So the antibody that's like it attaches, it's the first one, and it triggers an immune cell, a B cell, to make more antibody of the same kind. Okay, so if we only have one specific cell and one specific antibody, then it's going to just trigger that one cell to make a bunch more of whatever was invaded. And it also triggers that same B cell to replicate just madly, just keep going, turn them out, turn them out, turn them out. And when it gets to that stage where it's starting to produce antibody, then it's called a plasma cell or a plasmacyte. Cyte meaning cell. So basically the same thing. Okay, so, oops, where am I going? Sorry, backwards. Okay, so here we have in the upper left, we have an antigen and it binds specifically to that virus. But as I mentioned, there may be thousands or even millions of antigens of that same virus, and one is not going to take care of it. So this antigen bound to this antibody causes the antibody to send a signal to the cell that produced it to make billions more antibodies. Okay, so this is a plasma cell churning out antibodies, and it's very specific. And then, of course, it replicates again, and it replicates again, and each of those cells is now sending out more and more antibody. So oops, um, when you get a cold, um, like I said, the innate immune response is just instantaneous, right? Your mucus starts working. You're trying to sweep it all away. Like, hurry up, let's try and get this out of here before it gets, any worse. But obviously, it's overwhelmed and it can't do that. So you get an antibody, and the antibody is going to cause more of its own kind to be reproduced in the cell that made it to get reproduced. So after a certain period of time, like 2 days, 3 days, you're getting enough antibody to attack all of that cold virus that got into you. It still takes a little bit of time for all that binding and all that clearing, but after about 3 or 4 days, you start to feel a little better. By day 5 or 6, you really are feeling pretty good. And that means that your immune response has cleared the cold from your body. And then eventually you're well. Again, beautiful system. All right, so active immunity. So once the antigen is cleared, most of those immune cells or those plasmacytes are going to die, right? You don't need them anymore. You've got that virus. It's all cleaned up. You've gotten rid of it. The phagocytes have taken care of everything. However, a few cells that make that particular antibody hang around because they say, we saw it once, maybe it's coming back. We don't know, but we're just going to hang out anyway. So it oftentimes is just one cell. Usually you start out with, I don't know exactly, but let's just say 50. And then over time, it's going to, some of those cells will die and others will remain. And, eventually you get down to just a few, just a few cells. And because of that, because of aging, that's why some of older people don't have the immune system that, say, a 30-year-old or a 20-year-old has, because those 30-year-olds or even 40-year-olds, they have seen lots and lots of different antigens. They have made lots and lots of different antibodies. But as they age, those cells start to die, and the cells that make antibody that you've seen before, even, they're going to die. And so that's why elderly folks have reduced immune systems. All right. The antibodies, though, they stay around even longer. So theoretically, if you do see something again, like let's say after 20 years, you're still able to mount a rapid immune response. And as I mentioned down here, this is why moms don't always get their kids infections, because they've seen this antibody before, and once you've had a particular virus, a cold virus, you can't get that same virus again. Isn't that amazing? There are thousands of different types of cold viruses. So if you get a cold again, it's a different virus. Might be only slightly different, like might have, like it has three different, whatever, proteins on it. And this time it has four, it still feels the same. It causes congestion and, achiness or whatever, but it's different. When you've had it once, you don't get it again because you have antibodies and plasma cells that are hanging around. Now, last week, no, 2 weeks ago, I'm staying with my daughter and my three grandchildren. So 2 weeks ago, two of my grandchildren started getting a cold. And I'm like, oh, great. Am I going to get it? Am I not? Is this a new one? Is this one I've had? So what happened was after about 3 days, I started feeling like, am I getting something? Maybe it was the interferon. Maybe it was just a little bit of stuffiness. But then like within 12 hours, I didn't feel anything. So what happened was I probably got a healthy dose of that cold virus because, they climb all over me. And so, it took not very long, but longer than if I had just seen that virus like a week ago. I'd probably seen that virus, 5 years ago, 10 years ago, whatever. And so it took about 6 or 8 hours for my antibody system to create enough antibody again to get rid of it quickly. I mean, I never felt really sick. I just knew, okay, I've been infected with it. And, being a microbiologist, I can tell. I know pretty quickly, not trying to be cavalier or anything. It's just, I know what's happened. So anyway, you will too now. And so hopefully you won't get things again unless it's something new or it's mutated. All right. So specific immunity is called adaptive or active immunity. Adaptive and active, both meaning we're making something, we're adapting to this challenge. So here we have a little diagram. At the top, we have the antigens, and that's the bacteria. In this case, it's a bacterial cell with a few little protein coats or a capsule or something on it. And then we have three different B cells that it comes in contact to, and only one of them has the right kind of antigen binding site, and that would be number 2. Okay, so number 1 and number 3, they can't bind with that. They'll bind with something else if we ever come across it. But number 2 is the only one that can actually bind with that. So once B cell 2 sees that antigen, then it's going to, again, send out some chemical responses, and it's going to tell its plasma cell progenitor, the cell that created it, that it needs more it needs more antibody, send out more of the same antibody, and then replicate yourself so that those cells can send it out too, just in case we have a whole bunch of this antigen. And that's what's happened. So we have proliferation and maturation of those same B cells, and then all of those produce memory cells. And then you can see over on the lower right-hand side, there's a few cells that are called memory cells. And those are the ones that hang around for lots and lots of years. So this is called active immunity, as I mentioned. And there are 2 types of active immunity. So the most common is naturally acquired. So that's the immune response against antigens that you encounter in daily life. And believe me, you're bombarded with antigens. You're attacked with antigens daily, all day long, all night long. It's like you're breathing in something that's an antigen, you're might be digging in the garden, you might be touching food. You're just bombarded with antigens all day long. Okay. And that's naturally acquired. So you've made antibodies to all of those substances that you've encountered since birth, and you have memory cells to those so that they don't infect you. Okay, so that's naturally acquired active immunity. You're doing something. Then there's artificially acquired, which means you're still going to create a response, but it's not naturally acquired. So in this case, this is what a vaccine is. Okay, so they take, let's just say, let's take COVID because that's the most common. Now, COVID is a virus, and it's a capsular virus, but it has spikes, protein spikes on it. And so the antibody, or excuse me, the vaccine that they've made has a portion of that protein spike, just a little bit of it. And then they reproduce just that protein spike, and they put it in a vaccine, and then they inject the vaccine, the little protein spike, into you. And so your body says, antigen, 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 alert, alert. And so it starts, whatever antibody you have that's specific to that antigen will bind to it. It will agglutinate, and the phagocytes will come and take it away. In the process, you're also making more antibodies, more plasma cells or B cells, and you're making tons and tons more antibody. And then some memory cells will hang around because your body's like, we saw this once, it just might see it again. Let's be careful here. So then you've got your COVID vaccine or your coronavirus vaccine, and and then you're walking in the grocery store and someone coughs on you, and lo and behold, a bunch of coronavirus enters your nasal cavities. Well, your body gets to work immediately. It says, ding, 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 ding. Here comes the coronavirus. We've seen it before, guys. Let's get to work. And so all of those plasma cells start reproducing again. They're pumping out antibodies. And then those antibodies take care of that coronavirus before it can grow, before it can infect you and make you sick. And you don't feel a thing. But all the while, inside your body, all of this process is going on that's going to keep you healthy. So the same is true for the vaccines you got as a child: measles, mumps, rubella. You probably, no, I don't think you got polio. You might have, but I don't think so. All of those, and diphtheria, tetanus, lots of different childhood vaccines that they give you to protect you from illnesses that are very common. And well, they were very common, but now with vaccines, they have become much less common. There's much less death and much less suffering among children. So I'm old enough to have had some of those, and I can tell you that I am very glad they have vaccines now. I have no hesitation or had no hesitation in getting my children vaccinated because it's such a wonderful way to have artificially acquired active immunity. Now remember, natural acquired is when something bites you, something infects you, you develop it. It's naturally acquired active immunity. Artificially acquired is like a vaccine. It's put in by somebody artificially, but then we do still make the response to that particular antigen. All right, here's an example of it. Artificially acquired response to antigens introduced via a vaccine. In this case, we have the baby there who's getting the vaccination, and it's going to stimulate the immune system. It's going to have B cells that are churning out antibody. And then the next time the antigen is present, you have a super quick antibody reaction. So then it takes care of it before you even feel a thing. All right, so what are some types of active immunity? So these are artificially acquired active immunity. So there are different types of vaccines. So there are live attenuated microbes. So getting a vaccine, which is live, means that bacteria, that virus, whatever, is still alive. Okay. They don't use these as much anymore, but they are important because if you inject an entire microbe, you're going to have antibodies to all kinds of parts of that live virus. You might have antibodies to the cell wall. You might have antibodies to the flagella. You might have antibodies to the cell membrane. You might have, so you can see that the more sites that you can make an antibody response to, the stronger your immune system is going to be. However, in this case, in most cases, we give live attenuated microbes. And attenuated basically means we've changed them somehow so that they're not infectious or they're less infectious. So in this case, the tuberculosis vaccine, we used a weakened mycobacterium. So it's not really able to cause an infection in us. It's the same thing, but it's just been weakened. The concern with this type of vaccine is that it might revert to the more virulent form. When it gets into your body, we just don't know what's going to happen. In most cases, these vaccines are used infrequently now. They were the first type of vaccine to be produced. You might remember Edward Jenner and his vaccine using cowpox. And so he used live cowpox to prevent against live smallpox. So that was the beginning. But now, like I said, we use some different forms. So the second type is killed or inactivated microbe. So this is like you take E. coli, you kill it, and then you inject the entire organism into, or you put that into a vaccine, and then it's less effective, but it still works. You're not likely to get an E. coli infection by being injected from in a vaccine. Very often, though, you're going to need booster shots because it's killed and you're not making as many antibodies to all the different parts of that organism. So in this case, most microbes are, heat or chemically killed. Then we have toxoid vaccines. So there are some organisms that make toxins, and one is tetanus. Okay, so the tetanus organism is we don't want to inject it, but it makes a toxin. And so we can create a vaccine that has an inactivated toxin. And we put that into a vaccine. And then after a person has been exposed to that, as soon as they see that toxin again, they're going to make a bunch of antibodies to it, just like before, all those plasma cells, all those antibodies. And then they're going to create that response, and it's going to kill it's going to attach and eliminate any toxins that are able to get into your body. Okay. And the way they make these is they will inject toxins into, most commonly, I think they use sheep now, but they'll inject a small bit of toxin into it, and then the sheep is going to make, and we use many sheep, of course, it's not just one sheep, but then they'll draw some blood from that, they'll spin it down, it'll be able to just withdraw the toxin portion, use that in a vaccine. So we've had a couple of different vaccines. Now, the newest vaccine, and this is what they use for coronavirus, was DNA or RNA vaccines. So the gene which causes the virulence, which is the infectability, is cloned. So it's recreated or reproduced. Then that little bit of gene from the coronavirus is put into a non-virulent or non-disease causing microbe, bacteria or a virus, and then placed into a plasmid, which we haven't talked about. Well, we did talk a little bit about plasmid, which expresses the gene inside the cells of its recipient. Okay, so this is it simplified. So here's the virus. The virus is the long green looking snake-like thing, if you will. So there's different genes on this, and they code for different things. So starting from the left, there are certain genes that code for the protein spike on the virus. That's the capsule that surrounds the virus. Then in the middle there, we have reproduction. Those are the genes that code and tell the virus how to reproduce, how to take over your cells and reproduce. Then there's another section here, which I've labeled insert into host DNA, which is, it doesn't really have as much of a function. It's just a very small snippet of something of DNA that has a specific sequence. And that is what goes into the host, the host DNA. That's how it gets in there. And then there's another bit that can dissolve the nuclear membrane. So the virus has several ways, several genes on it that tell it how to operate in your cell. So what they'll do is they'll take a small bit of that DNA from the virus. And in this case, I use the protein spike. So the protein spike isn't going to hurt you in any way, shape, or form. They take a little bit of that DNA from that particular virus. So it has a very specific DNA sequence. They reproduce it. So you've got tons of that little protein spike DNA there. And then that you're going to be the, after you're vaccinated, the RNA spike protein code is now an antigen. So that little snippet of DNA goes in, and then you see it and you create the little yellow antibody there that fits it exactly. And the whole process starts, creates more, your body creates more and more and more and more antibody. It calls in the plasma cell, tells them to reproduce like crazy, and they produce more antibody. And then you are now vaccinated. You're vaccinated against the coronavirus without ever having to be exposed to the coronavirus. Now, the difference between the RNA and the DNA viruses, some people are really scared about them, but it's basically, instead of taking the little protein spike chunk from the virus, actually taking that, it's just taking the DNA that says, this is how you make it. Okay. So you're not getting any DNA into you. You're not, you're not the DNA. Okay. So when you get a virus, the virus takes over your own cells and it uses its DNA to tell your cell how to now operate. So it takes over, right? By using this little tiny piece of the virus, you're not getting any instructions put into your DNA. You're not getting anything into your cells that is harmful. You're just getting a little piece. It's like dirt, if you will, but it is DNA, it is RNA or DNA. So little piece of it, and then your antibody response just goes crazy, and then you never have to get coronavirus. All right, so this is just a little bit of an example. I'm sorry if I rambled on there. I'm trying to explain it, and I know it's a little bit difficult. So this is the coronavirus. That's it. That's it in electron micrograph, and then the one below it is kind of a continuation via a graphic or a little cartoon. So you can see the cross section, or even the big one. It has protein spikes. Those little red things are protein spikes on it. And then the white part is the capsule. So it's encapsulating that DNA or RNA of this particular virus. And you can see the strand right there inside the protein code of the virus. So in this case, when they're making the vaccine, they're going to take a little snip of that DNA, and you can see it on the upper right hand side, just that little snip. Then what they're going to do is, this is a DNA, this is a plasmid from a bacterial cell, and they're going to be able to open up that plasmid, separate it a little bit, and then insert that DNA or RNA from the virus. Okay, so here we see it. Here's on the upper left is the created plasmid. It's now in this bacterial cell. You can see the plasmid is smaller than the chromosome, which is the larger circular DNA part. This bacteria has a cell wall and it has a flagella, just so you remember. Then we're going to grow a bunch of those bacteria, right? Bunches. You can see all of them have that plasmid in it. So they're reproducing. And then what we do is we take all of the product from those cells and we filter it and eliminate any products that are not what we're looking for. So we're looking just for that little bit of DNA or RNA, which is represented here by the blue. And then we take that little bit of DNA or RNA and we put it into your vaccine, and then it goes into your arm. Okay, so it's simple as that. There's no There's no genetic engineering that's going to take over your body, or it's not going to change any of your DNA, or, they're not monitoring your genome or anything like that. So a lot of misconception around that. But anyway, hopefully now you understand it a little bit better. All right, so reviewing types of vaccines, there's live attenuated, and those vaccines I mentioned were tuberculosis, and that uses a weakened form. We also have the oral polio vaccine. Now, when my daughters were young, they got a couple of drops, and that was live polio. And in that case, they're like, be careful changing diapers, make sure you wash your hands, because if you ingest it, you might be getting polio. But it doesn't affect the infant in that way because they have such a low dose of it, but when they have it, they may be making more of it. It's anyway, let's not go into that. Anyway, live attenuated, you're giving an alive product. So tuberculosis, the oral polio vaccine. They also used it for measles, yellow fever, and rotavirus. I'm not sure if all of those, I'm not sure if measles is still a live attenuated virus, but it may be. Then we also have killed antigens. So pertussis is a whole, pertussis is a bacteria. So they take the whole bacteria, they reproduce it, they kill it, and then it goes into the vaccine. And then also the inactivated polio virus is what they use, I think, more commonly now. Then we also have a subunit vaccine, and that is just a little portion of the organism that's going to infect you. So in this case, what I have listed here is acellular pertussis, Haemophilus influenza. All of these are and pneumococcal and hepatitis B. Those are all bacteria, and they just take a tiny little fragment of that bacterial cell, and then they reproduce that and make it into a vaccine. So that just a portion of it goes into your arm. And then the toxoid. Remember, that's inactivated toxin, goes into your body. They use that for tetanus and for diphtheria. Thirsty. Get thirsty from talking so much, sorry. Okay, so specific immunity, we also have what's called passive immunity. We talked about active immunity, and that's where your body is making an immune response. So you see an antigen, you make a response, now you have active immunity. We also have something called passive immunity. So passive immunity, we do not make the antibody or the immunity. The immunity is passed on to us. So passive, passed. So we have naturally occurring. So that's when antibodies in a pregnant mom cross the placenta or in breast milk. They're protecting the baby from anything the baby might see in the meaning antigens, in the first days and weeks of life before it has an opportunity to see and acquire any active immunity. We want to protect that little tiny thing from dying right away. So we have naturally acquired antibodies, and these are antibodies passed on by the mom. If they're not making antibodies, they're passed on by the mom. Then we also have artificially acquired, and this is where we receive antibodies from another individual. So I have a lot of stories to tell this, don't I? So long time ago, I was working in the blood bank or transfusion department as a medical technologist, and I got stuck by some blood, a needle, and it had blood in it, and they thought the patient might have hepatitis B. So I went down to the ER, and they gave me a big shot of antibodies of hepatitis B. So what that did is it bypassed my entire immune system. They injected a bunch of antibodies that if there were a few little antigens of hepatitis B floating around, this big dose of antibodies they gave me would find those and kill it. So I didn't need to make any active response. I had just gotten injected with artificially acquired antibodies. Okay, so passive immunity has two types. So does active. So active immunity has naturally acquired and artificially acquired. Active immunity, naturally acquired, is where we see an antigen and we make a response. Active immunity, artificially acquired, is where we get a vaccine. And because we get artificially injected, we still make an active response. Okay. Passive immunity, in the same vein, has naturally and artificially acquired. Okay. So naturally, the antibodies cross the placenta, and artificially, because I got an injection, you could get an injection of antibodies from somebody somewhere, and then you would never get the disease. I hope that's clear. So here's just a little diagram to help understand that. So natural, you get an infection, and this is active immunity. Natural is getting the infection, artificial is getting the vaccination. Passive immunity, natural is maternal antibodies, artificial is you may get antibodies for some disease that you accidentally got stuck with. All right, so this is a chart of kind of everything we've covered. So on the left, we have the global topic of immunity. We have adaptive immunity and innate immunity. In last lecture, we talked about innate immunity. In this particular lecture, we've talked about adaptive or specific immunity. We might have naturally acquired immunity, and we might have artificially acquired immunity. Okay, so just another way of looking at that to try and make that clear. Oh, and this is Paramecium parlor. Hey guys, check out my new hat. Oh no, get away, you've been marked. So the paramecium over there, you can see he has an antibody on it. And thus, Ned learned the dangers of accessorizing with antibodies. So we love them, antigens don't. Thanks for listening.